Hi, my name is Nathan Morkey, and today I'm going to talk about my final project, which is a big data case study in the life sciences. I want to begin by describing how information and data is encoded in all living organisms. This encoding is in the structure of the polymer called DNA. DNA is comprised of four unique individual bases or chemical subunits. These are adenine, thymine, cytosine, and guanine. And these individual subunits can be represented by four letters A, T, C, and G. This simple DNA alphabet is able to encode thousands of different proteins. These proteins form the building blocks of cells and run all the biochemical reactions that are necessary for sustaining life. The total complement of DNA in a human cell is referred to as the genome. The genome, comprised of 3 billion individual unique DNA letters, is organized into longer sequences called genes, and genes encode 30,000 different unique proteins in an individual human cell. The genome is a higher level of organization into larger structures called chromosomes, and each cell has a complement of 46 of these structures. As you can imagine from this, the analysis of DNA sequences is fundamental to every area of the life sciences, from modern cancer diagnostics to the study of very tiny microorganisms. In the last decade, the advent of a new technology called next-generation sequencing has dramatically increased the availability of DNA sequence data. In next-generation sequencing, a sample of DNA from an organism called the genomic DNA sample, which comprises its entire DNA complement, is fragmented into smaller bite-sized chunks or sequences. These smaller chunks of DNA then are analyzed in an instrument which can run millions of sequencing reactions in parallel. And what you have is a large collection of small DNA sequences called reads, which then can be aligned to a master reference DNA sequence composed of the entire genome of an organism that you're studying. Then, once you've aligned all your individual reads to the master genome reference sequence, you've reconstituted the entire unique genome sequence of an individual human being or a unique microorganism that you're studying in the experiment. The advent of NGS has created a significant challenge for big data analytics. This is well illustrated by this figure showing over the 2000s the dramatic increase in depositions of DNA sequences as shown in red compared to the increase in depositions of protein sequences as shown in blue and green. And an increasingly important strategy for analyzing these large DNA datasets is the implementation of MapReduce computational approaches, frequently using a Hadoop framework. This brings me then to the final project which I'm demoing in this presentation, and this is an implementation of SeqPig, which is a platform designed to analyze large DNA sequence files. SeqPig is, as you can imagine, an extension of the pig scripting language. It allows a user to write scripts that are translated into MapReduce programs without worrying about the fine details of the implementation of MapReduce programming. In the case of SeqPig, it extends standard pig by providing new functions for loading, saving, and processing DNA sequence data. My demo and process of installing and running SeqPig is comprised of a number of steps. First, since I'm running all this on the Amazon Web Services framework, I need to download and install Cygwin or another suitable SSH client so I can log into and access the master Hadoop node. Secondly, I need to set up a storage system, in this case Amazon S3, which can persistently hold SeqPig scripts for analyzing data, as well as the target DNA sequences to be analyzed, and text files that contain the results of analysis by scripts. Then, of course, I need to actually generate and run an EC2 cluster on the Amazon framework, and this cluster needs to have both Hadoop and Pig installed. Then once this is up and running, I use Cygwin to SSH into the master node of the cluster, and I can install SeqPig on top of the underlying Hadoop Pig framework. And once this is done, I'm ready to roll and run SeqPig scripts that I've written on DNA sequences that I'm interested in analyzing. This is an illustration of how I've set up data storage on the Amazon S3 bucket network. I have 4 buckets in this case: a SeqPig script, which stores the scripts to be used to analyze data; SeqPig input, which stores the DNA sequence files. In this case, I'm analyzing DNA sequences in the BAM format, which I'll describe. Then there's SeqPig output, which contains text files which are generated by the SeqPig scripts. And finally, SeqPig logs, which contains the logs generated by running these processes on the cluster. So this is just a status window showing a cluster that I have up and running. As you can see, it's running Hadoop and Pig. An important note is that SeqPig isn't compatible with more recent versions of Hadoop 2.0 or later, so I'm running Hadoop 1.0.3. Once an EC2 cluster is up and running, then it's straightforward to access it via SSH from Cygwin and go through a process of installing SeqPig. And in my demo, I'm grabbing, using wget, the SeqPig already compiled code from the SourceForge distribution. And then I simply just need to unzip the already compiled code and update some of the system variables, and then SeqPig is ready to run. The example I'm going to talk about in my demo is that of tracking DNA barcodes. In a lot of next generation sequencing experiments, because the instruments have such a high capacity, you actually are pooling data from multiple patients if you're in a clinical diagnostics lab, or multiple organisms if you're studying basic biology, for example, studying different pathogenic bacteria. So in this case, when you're multiplexing your samples, you add an additional step. After you fragment the genomic DNA, from each individual patient or sample, you add a specific small DNA barcode sequence. This barcode sequence stays on the fragments throughout the entire process of sequencing, and then when you're doing your final analysis, you can use the DNA barcodes to track the original patient or organism that sequences came from. What I set out to do was write a very simple SeqPig program that can take a large collection of DNA sequences that have been aligned to a reference genome but might come from different individuals, and fish out all the sequences that contain a particular DNA barcode. So to describe this script in brief, it first loads DNA sequence data using a function in SeqPig called BAM loader, and this is used for loading data from the BAM storage format. This storage format is a compressed binary format which contains DNA sequences that have been aligned to a master reference sequence, along with accompanying information describing the sequence quality and where it's located in the alignment. After I've loaded in my data, then I do a simple pattern matching using a regular expression to filter out all the sequences that contain my DNA barcode of interest. I then do another filtering based on the mapping quality to exclude low quality mapped DNA sequences, and then generate a data structure containing the sequence of interest along with additional identifying information. And finally, the script outputs this in a comma delimited text format, which can be read downstream by other programs. The running of this script then is very simple from the command prompt. You simply need to grab it from the S3 bucket using a Hadoop file system command, and then run it with various parameters defining the input and output destinations. And importantly, using a regular expression, the specific pattern of the DNA barcode that you're trying to fish out. This is an illustration grabbing part of the output of barcode.pig. This is a comma delimited text file, as I mentioned. As you can see, we've fished out a bunch of DNA sequences, and as shown in bold, they all have the ATC ACG barcode that we wanted to isolate based on. Then this text format can be sent downstream in the pipeline to other programs for further analysis. With this demo, I've identified a few advantages and disadvantages to using SeqPig to analyze DNA sequence data. The advantages include the fact that it's highly scalable and the way it uses the Hadoop MapReduce network, it's very readily applicable to large DNA data sets. And the files generated by DNA sequencing often are huge, on the order of a terabyte or more. Furthermore, it's very simple and easy programming, and you don't have to concern yourself with the fine details of MapReduce. And SeqPig is ultimately based on Java, so it's highly portable among different systems. SeqPig does unfortunately have some disadvantages as well. Because SeqPig uses very generic MapReduce algorithms that aren't optimized for a specific task, it might not be as efficient and fast as other bioinformatics programs which have been developed and optimized for very specific tasks in DNA sequence analysis. And furthermore, in the process of running this demo and testing out SeqPig, I found that some of its functions aren't properly implemented. For example, when trying to use one of the loading functions for the FASTA DNA sequence format, which is a very common format used in a lot of databases, I found that I could load in the data, but that the sequences weren't being properly represented on the Hadoop file system. So to summarize this demo in the project, what I'd like you to take home is that the rise of next generation DNA sequencing has really created a significant challenge for big data analytics in the life sciences, and that MapReduce programming platforms are potentially a very good solution to this problem, as they're very scalable for large data sets and large files. And DNA itself is readily amenable to MapReduce analysis, as you can easily divide a long DNA sequence into smaller, more bite-sized chunks for analysis. And then specifically for the demo that I've run, I've shown, and I hope I've convinced you, that SeqPig is a potentially useful framework for doing MapReduce analysis of DNA sequence data, although it does have some kinks and technical issues to work out that hopefully will be solved in future versions and future development cycles of this platform. Thank you very much for your attention.